Today we are going to have a discussion about pediatric brain tumors and how team science can be used to address this problem. Our speakers today are Drs. Don Coulter and Dr. Sid Mahapatra. Dr. Coulter received his medical degree from the University of Arizona and completed both his pediatric residency and Hem/Onc fellowship at the University of North Carolina in Chapel Hill. He joined Children's and Med Center faculty in 2008, where he's a tenured professor of pediatrics and the Edna Ittner Professor of Pediatric Hematology and Oncology. He maintains his clinical practice as well as his research career and is the director of the Pediatric Cancer Research Group and is the lead investigator for the COG grant at the Med Center. Dr. Mahapatra is an associate professor of pediatrics and appointed also in the Department of Biochemistry and Molecular Biology. He completed his MD and Ph.D. from Rosalind Franklin University and then did his residency and fellowship at Lucile Packard Children's Hospital at Stanford and has been with us since 2015. He is actively involved in brain tumor research and has published extensively. He's a chair of the Pediatric Brain Tumor Collaborative at UNMC and is funded by the Pediatric Cancer Research Group and the Team Jack Foundation to develop a brain tumor core facility that will bank pediatric brain tumors and generate animal models to fuel bench research. Thank you. Dr. Coulter is our first speaker.
Pediatric brain cancer is the most common cause of cancer death for kids 0 to 14 years of age. Approximately 4% of the budget from the National Cancer Institute goes to pediatric cancer, and a very, very small fraction of that 4% goes to pediatric brain tumors. And to sort of put the importance on team science, pediatric cancer research output over the last decade has not kept pace with pediatric and cancer research overall output and remains at less than 5%. So obviously something needs to be done. How did I get started in this project? Clinically. In clinic, when I was a younger physician, I saw a number of patients who came from Aurora, Nebraska, and I had no idea where Aurora was. Aurora is in Hamilton County. You can see there are 9,000 residents in Hamilton County. Aurora is the largest city. And these are 4 patients that I had the honor and privilege of knowing who unfortunately all died. In thinking about them, on the right, you see Jacob Peters, and beside him to his right, my left, you see Maddie Spellman. The two of them were best friends and are buried right next to each other in the cemetery in Aurora. So trying to understand why I was seeing all of these children and my colleagues were seeing these children from Aurora, I looked at a map of the Nebraska cancer data from the SEER database of the United States. And if you look at this map, you can see that Nebraska is a red state, which means it has a higher incidence than the rest of the country. It actually has the highest incidence in the Midwest, and it's one of the highest states in pediatric cancer incidence outside of the Northeast, which contains the highest incidence of pediatric cancer. And this data has been stable for a long period of time, and nobody knows why. When you look at cancer mortality in Nebraska, Nebraska has an incredibly high incidence of cancer mortality. Now, the way that pediatric tumors are treated is basically protocol-driven in every industrialized country. So this would lead you to believe that pediatric tumors in Nebraska not only have a high incidence, they have much more aggressive nature than tumors in other places, which leads you to postulate that possibly there are mutational drivers that are making those tumors more aggressive. But again, we wanted to understand the geographic diversity in Nebraska itself. We wanted to find out maybe what's the incidence in Hamilton County compared to other places. When you dig down and look at that map, this is what you see: no data. Because the numbers of pediatric cancer for a 5-year period of time are so low that all of those counties have censored data, meaning that it's not stable, so it's not going to be shown, with the exception of counties that have people in them. So we wanted to do something about that, and we went to the state and discussed with the state the idea that if we had resources, we may be able to understand better the geographic variability within our state. So that's how the Pediatric Cancer Research Group was founded. This picture reminds me that this is not a solo job. And when people try to do these things on their own, you will fail. It takes a team. And this is the first team that led to team science. Who you're seeing there are the Allschweds. They are a mother and father of a daughter who had neuroblastoma that began a letter-writing effort that led to Danielle Conrad, who's in the middle, who's the senator who put forth the first bill to give us resources. And then standing beside me is Bob Bartee, who was our government liaison. So I'm in this picture and honored to be in this picture, but I'm standing here because of a number of other people, some who aren't listed in this picture. John Sparks, Peter Kosha, Jill Beck, Bruce Gordon. The list goes on. So it is definitively a team effort. And I think what the state was interested in was both short and long-term goals. The short-term goal, identifying what the geographic variability in our state was to understand how we would better give out health care to rural populations. The long-term goal was how do we impact moving science forward in Nebraska? And that goal becomes critical because of data like this. This comes from the National Science Institute, and it shows that people getting their first R01 with an MD degree, it can take an incredibly long time. If you look all the way to the right of that slide, you can see that it can take until someone's about 42 or 43 years of age, on average, from when they got their MD degree. So the available workforce of people who can actually start doing the research that we need, they're getting older and older. And add to that the fact that it can take a very, very long time for people to receive their first R01 or NIH grant. So if you look at this slide, this is people who have either an MD or an MD Ph.D. And as you look to the right, we're getting further and further in the years, and you can see that it can take, on average, up to 14 to 16 years from the MD degree to get your first R01. We need to do something to shorten that. We need to do something to make it so that people have more time to do science in their career, and it doesn't take as long for them to get support from extramural funds. So how would we do that? What are the barriers that these researchers are experiencing? Well, identifying mentors who can help them through that. That's incredibly important in our department here in Nebraska because our department has mostly a clinical focus, which is wonderful when you're thinking about doing research because we really want to get it to bedside. But when you have a number of people who have a clinical focus, they might not have all of the available time to them to do the research side. Any research project is always going to start with sleepless nights and long weekends. But if you can have someone to help mentor you through that, that's incredibly important. Identifying funding. You need early funding to be able to do something. Developing preliminary data from that funding, and then establishing a research publication record. And as you see with Sisyphus, who's rolling the boulder, he can't get it up there because he's doing it on his own. You need a team. So that was the secondary aim of the Pediatric Cancer Research Group. And in developing the group, we were able to mentor young clinicians into having the ability to share their research with others. We were able to put people together in teams, College of Public Health people with clinicians to understand what the next steps might be. We were able to garner more philanthropic funds to continue to push the research forward. We were able to use those philanthropic funds to help very, very young members of medical school or Ph.D. students to be able to present their early research. And we were able to engage with the community and get to people in high school to show them the importance of science and how it can be a career. All of this while making sure that we engage our government partners like Governor Pillen, or we engage leaders at both Children's Nebraska and at the University of Nebraska Medical Center to make this go. But there's another side to this. So we've talked about pediatric cancer. Pediatric brain tumors are a very, very specific and difficult area to research. This is Team Jack. And as you can see Jack Hoffman himself standing with President Obama. Jack Hoffman was the young man who ran the touchdown and won the ESPY Award many, many years ago when he was with the Nebraska Cornhuskers for their spring game. Jack has a diagnosis of medulloblastoma. His father, Andy Hoffman, started Team Jack, and unfortunately, Andy Hoffman passed away a few years ago from a glioblastoma multiforme. 2 different types of brain tumors coming out of Holt County. We're not alone in Holt County. In Nebraska, in this 23-year period of time where we looked at all of the data, you can see that the incidence of pediatric brain tumors in Nebraska is climbing at a rate that exceeds the rate in the rest of the country. And that data is actually mature for the United States. This data will show you that as you look from years 2004 to 2019, the incidence of pediatric brain tumors continues to climb. It's climbing so much in the state of Nebraska that pediatric brain tumors are actually our most common diagnosis. Most pediatric oncologists will tell you that leukemia is the most common diagnosis, as referenced in the SEER data in the pie graph on the right. But in Nebraska, brain tumors are our most common diagnosis. This is data from the Central Brain Tumor Registry, which looks throughout the entire United States. This is the most up-to-date data. And if you look at that set, you'll find that Nebraska has the highest incidence outside of states that are in the Northeast or states that are in the Mid-Atlantic. So we do have an issue. As we try to investigate what the issue is, we try to figure out what makes Nebraska different from other states in the Midwest. And by far, the Ogallala Aquifer is a wonderful resource that we have that supplies 85% of our groundwater. But that aquifer is naturally contaminated with heavy metals like nitrate and uranium and arsenic. And it's also contaminated with agrochemicals that are required to grow all of the food that we make in Nebraska, like atrazine. So we have teams that are looking at counties that have a high level of atrazine, which you see in the top left, high levels of nitrate, which you see in the top right, and then comparing that to the incidence of pediatric brain tumors. And just for example, if you look at Holt County, you can see that it has a high incidence of both atrazine, nitrate, and you can see it has a high incidence of pediatric brain tumors. Those maps are, for most places, superimposable. So that's why team science is so incredibly important. Team science not only find out why we have such an issue with pediatric brain tumors in Nebraska and understand if our environment is a part of that issue, but also understanding what can make those tumors more aggressive and give us the high mortality rates that we have in Nebraska. If you have any more interest in talking about the environmental exposures or any of the environmental research that we're doing at the College of Public Health, I urge you to register for our free symposium. We're taking this sort of data set on the road, if you will, and we're inviting representatives from Iowa, Missouri, Kansas, and Wisconsin. And they're coming to Nebraska in July to meet with our College of Public Health, led by Shinobu Watanabe Galloway, to discuss ways that we can work together to look at incidence rates in the Midwest and talk about some of the other things that are important when it comes to pediatric cancer care, whether it's financial toxicity for families, whether it's access to care. We'll be doing that in July, and you're all welcome to come. But I would like to hand this off to Dr. Mahapatra, who has been incredibly successful in developing a focus in pediatric brain tumors and team science within the Pediatric Cancer Research Group.
Thanks so much, Don. As you can see, Don has done some incredible work to lay the groundworks for the work that I'm going to introduce in the next few slides. So I'm going to bring you up to speed on the Pediatric Brain Tumor Collaborative and how this collaborative represents one of the team sciences approaches that we're using at the University of Nebraska Medical Center to tackle the very problem that Don has introduced with respect to cancer in our children in our state. Now, the Pediatric Brain Tumor Collaborative was started in September of 2020, right in the thick of the pandemic. Because as you can recall, during that time, we were all kind of sent back into our houses, locked down, and only essential personnel were going to work. Many of us in the lab were actually not allowed to go to lab. And so this was a time where I kind of started to think about, what have I been doing till now, and how is that impacting the incidence of these tumors and the understanding that we have of these tumors, and how can we move that needle forward faster? And that's when I thought that it would make sense to bring a team of us together to actually work on this. And I knew that there were multiple different labs at UNMC that were doing incredible work that I had been introduced to through the Pediatric Cancer Research Group, and I figured, why not bring them together? And so we started the work then of developing a multidisciplinary group of clinicians and scientists that now makes up the Pediatric Brain Tumor Collaborative. So first and foremost, we've got our two oncologists that lead this, Dr. Don Coulter and Chiralji, who's our neuro-oncologist. We've got our three neurosurgeons who graciously joined our team, including Dr. Klug, Dr. Salehi, and Dr. Lai. And then we have 8 other basic scientists that have, and I put myself in that group, that have labs that are tackling different types of brain tumors and different aspects of those brain tumors in order to make this very robust group of clinicians and scientists that are working together. One of our earliest meetings, really, we wanted to talk about, we want to start off on the right foot. What is the vision of our group? And really, it's aligned with the vision of Children's Hospital and Medical Center, which is to improve the lives of children with high-risk brain tumors. Our mission really was taken from this group of high-powered researchers, which is really to capitalize on complementary, and that's what's important, complementary biomedical expertise within the CHRI and the greater UNMC campuses to collaboratively advance our understanding of high-risk brain tumors. So as Don mentioned, we are not able to do this alone. So how can we access the different expertise that are available throughout the campus to help us with this major public health problem? And our goals were to really allow for a free exchange of ideas that may result in better quality research, to provide a panel of pediatric brain tumor experts that are both from the clinical side as well as the basic sciences side, who may provide peer review for our future projects, manuscripts, grants, and really to eventually procure multi-PI national funding and potentially get us to the stage of clinical trials for therapeutics that we're testing and developing within the lab. We also have a website, and that's the web address for that. So what is our primary research focus within the PBTC? So right now we are focused really on looking at high-grade gliomas as well as pediatric medulloblastomas, which were introduced to you in the prior slides. And here's a smattering really of all the work that is going on and how these things are complementary. So as you can see, there's no one single circle that encompasses all of these aspects. You see all of the different investigators highlighted there, as well as the work that they're doing. So Dr. Mose, who's in the College of Pharmacy, he's looking at tumor targeting using fluorescent dyes so that once that tumor is resected, fluorescence can help us see things like cancer stem cells or other areas of tumor that are not showing up within the MRI. Dr. Rosino is looking at SOX2, which is really a marker of dedifferentiation that he is showing is a marker of aggressiveness in pediatric brain tumors. Dr. Bucket is looking at the DNA response mechanisms and how can we leverage different ways to mutate and deregulate those to treat our tumors. Dr. Chaturvedi is looking at MYC inhibitors, MYC being a high-risk marker within brain tumors. Dr. Trippier is a chemist. He's a medicinal chemist who's actually developing synthetic drugs and inhibitors for multiple different deregulated proteins within aggressive tumors. Dr. Mahapatra is an expert that's looking at nanotechnology and how can we use nanoparticles for the delivery of these novel particles that Dr. Trippier is developing. I'm trying to look at pathophysiology and using that pathophysiology to drive treatment by really looking at iron imbalance and how can we leverage that, which I'll introduce towards the end of this presentation. Dr. Ray is looking at the JAK-STAT pathway, again, another pathway that's very commonly deregulated in aggressive tumors. And finally, Dr. Salehi is bringing his surgical expertise to the forefront and looking at how he can collaborate with Dr. Mose to use those fluorescent labels within his surgeries. Now, there's no point in having a team if we don't collaborate. And this is what happened as soon as we started this. Within the first year, all of these collaborations started to take off. And all we had to do was bring these people together, and they figured out how to use those complementary expertise to move their agendas. And it became a team agenda. And so I want to highlight 3 different teams right now that are working to move our work forward. So first, as I had introduced, Dr. Mose as well as Dr. Salehi, they're looking at fluorescence-guided surgeries for high-risk gliomas. They right now have hired folks onto their teams, and they're kind of moving their work forward. Dr. Mahapatra and Bucket are figuring out how to target the FACT complex, again, a DNA repair mechanism that is deregulated in many of these cancers, and how to usually use that to potentiate chemotherapy and radiation therapy so that you can kill those cancers faster. They have actually a joint R01 submission that's under review. Dr. Trippier and I are looking at a lead compound that he discovered that is really very effective in killing aggressive tumors. We've designated it B7H3NI1, and really we're trying to see how it is. We are actually seeing very good results of it killing cancer cells. Now the question is, how is it doing it, and how can you leverage that and bring that to the clinics? We have a joint DoD grant submission, as well as a Ph.D. student thesis project that is underway, and we've published some of our initial review findings within an article. Now, this is a group that is quite aggressive at the bench, and they're quite productive. So this is really the publication track record of this group since we started in 2020. And really what I want to highlight here is in the last 4 years, there's almost been over 100 publications put out by this group, with almost 56% of those being first and senior author publications. So these guys are very serious, very committed, and are quite prolific. And I'm very humbled to be a part of this group. Now, those were individual research articles coming out of their labs. We also together worked on a collaborative review that we put out in 2021 on medulloblastomas and really the subgroup-specific diagnostic differences in these. It was an incredible opportunity for me to work in a team sciences way of getting something like this out there that brings together kind of our knowledge and our expertise in the form of a review. Aside from just having our work out there and disseminating it, we obviously have to have outside funding as well to do the work that we do. And again, it's very humbling to be a part of this group that has submitted over 100 grants to intramural, extramural, national, and foundation level sources, and almost 43% of those are funded, which is a pretty high number. And what I'm highlighting here is the investigators within this group that either had prior or have current NIH funding, so really at the national forefront within this PBTC group. One of the deliverables that we've been able to bring from this PBTC is to generate this core facility. When I first started within the field, one of the biggest hurdles that we had, or that I had, was to figure out when I'm studying these tumors to understand these tumors. And to understand these tumors, I had to have access to these tumors. And I really had to go kind of door to door to the pathologist and the neuro-oncologist to ask them if I can take a piece of their tissue so I could study it better and understand it better. And I figured instead of doing that, within this brain tumor collaborative, if we had a core facility that was responsible for that, it would make it so much easier for new investigators and established investigators to kind of, again, start those investigations without that obstacle in place. So this core facility was funded by the Team Jack Foundation and the Pediatric Cancer Research Group back in 2022. And one of our first steps was to obtain an IRB that would allow us to procure these tumors from the bedside during the surgeries. And Dr. Grace Lai was instrumental in helping us get that IRB approved. And so we are ready now to procure pediatric brain tumor tissues during the time of surgery. And that's, again, the neurosurgical team as well as the pathology team are closely involved with that work. The other issue, though, is once you have those tissues and you learn about them and you kind of do your analyses, you have to have a way of testing your therapeutics, testing the signaling pathways that you're trying to target. And for that, you need animal models, especially in brain tumor research. And those are difficult to generate. They take a long time, and they have a very high kind of learner time. So what we've done is hired a tech who has expertise in animal modeling, and we're teaching her how to generate specifically pediatric brain tumor models, and then deploying that expertise for all of our investigators to facilitate their work. And finally, we actually had the privilege of working with a few different organizations from across the country, Nemours Children's, Nationwide Children's, and Dell Children's, on the diagnosis of a patient with medulloblastoma that really didn't fit the book, if you will. The child came in with what looked like a very low-risk tumor and was found to have a very high-risk tumor based on the work that we did within the core facility. So we're actually moving outside of just Nebraska and UNMC and helping other facilities manage these patients. So it's really exciting for us to have that opportunity. And this is just an example of what I had done when I first started, which is to bring all these tumors together. So this is a retrospective study wherein I took all of the tumors that we had over the 20-year period that was just medulloblastomas, banked them, did transcriptomic analysis on them, and published all of that online for people to have available. And this is kind of what we'd like to do in a model for the prospective work within the pediatric brain tumor core facility, so that any investigator will have access to a nice data set that they can start to ask their research questions through. And so I feel the future really is to now grow this core facility to then try to have some national impact through clinical trials and multi-PI grants, and hopefully that's going to be the fruit of the work that we're catalyzing by this team. And really the point here is to try to test and generate novel strategies to target these aggressive tumors so we can really move the needle to helping these children that have devastating conditions. And the ultimate goal really is to improve the lives of children with high-risk brain tumors. So in the last section of my talk, what I wanted to do was I wanted to highlight one of these novel strategies that we have discovered in my lab to target aggressive tumors. So I'm going to talk specifically about pediatric medulloblastomas. The pediatric medulloblastomas are the most common malignant brain tumor of childhood. They're subdivided. Well, first of all, they are located within the cerebellum or the posterior fossa, and there are multiple different subgroups, which I'm going to get into in a second. In order to make the diagnosis, we do multiple different things. We expose the tumor to multiple different tests that includes DNA methylation studies, solid tumor fusion analysis, next-gen sequencing, as well as germline mutational analysis. And this is in order for us to break it down into its 4 molecular subgroups, which are illustrated here. The circles represent the frequency, so the larger the circle, the more frequent that tumor is. But I want to highlight the most aggressive of these tumors, which is the group 3 tumor, which has the poorest prognosis, because it presents with high MYC amplification and metastases at diagnosis, which really makes it hard for these children. And those patients with group 3 tumors have the worst survival. Now, although there's a robust understanding about the molecular subgroups, these medulloblastomas are still divided actually into 2 clinical subgroups, either an average risk or a high risk group based on the age of the patient, based on whether the patient has metastases at diagnosis, and based on how well surgical resection has removed those tumors. And so there's a disconnect now between what they look like from a molecular substance and what they are actually treated with as far as chemotherapy is concerned. And so that divides them up into these 2 groups that undergo surgical resection, radiotherapy, and chemotherapy. And so because the molecular characteristics are really not being addressed through this approach, you see this big difference in survival between patients that have the most infrequent tumors, have the best prognosis, and patients that have these group 3 tumors by 30% incidence that have the worst prognosis. And that's really the point of what we're trying to do, at least within the PBTC as well as in my lab, is that we've identified this knowledge gap. What we have identified is we understand the SHH and the WNT tumors. We understand their pathophysiology. In fact, they are named for those signaling pathways that are deregulated. But we don't know that for the non-SHH, non-WNT, group 3, and group 4 tumors. We also have mouse models to test these novel therapeutics that have now been accelerated for these groups. We don't have that for our non-SHH group 3 and group 4 subgroups. And what that has led to, just to put numbers, is that the survival profiles are vastly different. If you look at the WNT and SHH groups, almost up to 90% survival. But if you look at group 3 and group 4, some of them less than 50% 5-year survival. And so the long-term goals of my lab is really to uncover deregulated molecular pathways that can trigger aggressiveness. What makes these tumors aggressive? Why are they aggressive? And once we understand why they're aggressive, how can we target them? How can we generate novel treatments that can potentiate the chemotherapy and radiation therapy that we have to help address and effectively manage these tumors? And so to do that, we went back to those tumors that we banked. Now, what is shown here is actually a different public bank of tumors, and what I'm showing is something called single sample gene set enrichment analysis, where every single one of these hash marks represents a patient and represents the gene expression profile of that patient in this pathway. Blue being low expression, red being high expression. And what jumps out at you is this thing on the top called ferroptosis, a pathway. And so I'm going to explain in the next few minutes what ferroptosis really is. Ferroptosis is a form of programmed cell death that occurs due to iron imbalance. To understand ferroptosis, we have to understand how the cell manages iron. Now, especially rapidly dividing cells, generally all cells need iron. Rapidly dividing cells need it even more. So iron is something that cancers are exquisitely sensitive to and need. So when we start off with, what we have is iron in the 3+ or ferric state being picked up by the transferrin receptor and getting into the cell. Once it is endocytosed into the cell, it joins within this lysosome where the acidification and the work of STEAP3 converts it from the ferric state to the ferrous state. And that ferrous state is what is picked up within different proteins and allows those proteins to be functional. But if you have the ferrous iron simply available within the cytoplasm, well, it can spontaneously go back to the ferric state. And that ferric state is a highly oxidizing agent and leads to this generation of ROS molecules which attack the lipid bilayer, leading to lipid peroxidation, and that then triggers ferroptosis, which kills the cell. Ultimately, that's what happens. Iron imbalance kills the cell. And so generally, ferroptosis really should be tumor suppressive, and it is. The programmed cell death that occurs through ferroptosis is a tumor exposes a tumor suppressive phenotype. So that's curious that medulloblastomas, I just told you, are very aggressive tumors. Why do they have such a high degree of ferroptosis? Wouldn't that make them less aggressive? Well, we looked at those tumors, okay, and we've divided them up into those that had high versus low ferroptosis activation. And what you can see with survival is those that have low activation actually have worse survival. This is not subgroup specific, right? These are all medulloblastoma brain tumors. So then we asked the question about the more aggressive subgroups, which is group 3 and group 4. And what you can see is group 3 tumors have the lowest ferroptosis scores. That suggested to us that ferroptosis evasion may be a mechanism that is used by these aggressive tumors to be aggressive. More importantly, though, now that we know that there's a pathway involved, there's hundreds of genes within a pathway, hundreds of proteins that are involved. Which proteins should we be targeting? How do we figure that out? So here you're seeing RNA transcriptomic analysis, which means you're looking basically at the expression of genes, or proteins, if you will, within certain patients, right? So each box represents a patient. Red is high expression, blue is low expression. These data here are generated from normal pediatric cerebella, and these are from tumors themselves. And again, each of these is a single patient and looks at the expression of that protein in that patient for that subgroup. And what you can see is a lot more red on your right, a lot more blue on the left. So these are all of the proteins that we found within those ferroptosis pathways that are upregulated. And they include things like the TFRC, or transferrin receptor, which I introduced a slide ago, STEAP3, also introduced a slide ago, a bunch of ABC transporters, which I'll come to, which are mitochondrial iron transporters, and then a transporter called mitoferrin, which brings iron into the mitochondria, and a transporter called ferroportin, which exports excess iron from the cell to the outside. And so this seems to me like an enriched pathway with an aggressive tumor. Maybe this is where we need to focus. And when we looked at this deeper, one of the most upregulated pathways, okay, so we looked at the actual gene expression, we looked at the single proteins, then we say, hey, how do these work together? Which pathways are upregulated? I spoke to you about the ferroptosis pathways. Within that is another pathway that's upregulated called the iron-sulfur cluster assembly pathway. So iron-sulfur clusters are found in DNA polymerases, primases, helicases, electron transport chain, everything that's critical for any cell to survive, but really critical to a tumor's way to proliferate. ABCB7 is a principal iron-sulfur cluster exporter found within the mitochondria, because these iron-sulfur clusters are made in the mitochondria and then used in the mitochondria or exported. So that's really the organelle of importance here where ABCB7 functions. And when you look at our tumors, here is, again, expression analysis. The stars represent the fact that group 3 tumors specifically have the highest expression of ABCB7. We now may be onto something. We went and looked at the survival of these patients, and this is just of patients with the aggressive group 3 tumors. What we found when we compared those with high expression of ABCB7, to those with low expression with ABCB7, you see a dramatic difference in their survival. Patients with high expression of ABCB7 have much worse survival. And so we began to focus our attention on patients on this protein. So we wanted to first show that ABCB7 is actually upregulated in our tumors. And so this is normal cerebellum, and this is a group 3 tumor, and you can see a lot of red here, which is basically staining for ABCB7. And so, yes, there's a lot of ABCB7 in our tumors. We've proven that. COX-4 is a mitochondrial protein. This is in green. And when you merge them together, what this picture shows you is that ABCB7 is highly expressed within the mitochondria of group 3 medulloblastoma tumors. Okay, so we found a protein. So now what do we do with it? Well, in the lab, we either upregulate or downregulate. We either silence it or deregulate it to see what does it do to our normal cells or our cancer cells. And so that's what we did. We used 2 different ways to silence the expression of ABCB7 in our cancer cells. And really, the output should be that these cells die, right? And that we trigger ferroptosis, hopefully. So in order to figure that out, again, let's go back to the pictorial. We need to show iron imbalance, we need to show oxidative stress, and we need to show lipid peroxidation if our hypothesis holds that inhibiting ABCB7 would lead to ferroptosis. So first, what did we do? We silenced ABCB7 in our cancer cell. Okay, that's a normal cancer cell with very little green fluorescence, suggesting that there's not much iron, right? So mitoferrogreen stains for mitochondrial iron. When we silence ABCB7, the cells light up. That means there's iron overload. This is a positive control. And then what we do is we then expose the cells to deferoxamine, which quenches iron, just to prove the point that it is all iron-mediated and not something else. So we've gotten step 1. There is iron overload if you inhibit ABCB7. Step 2, we looked at ROS. Again, we looked at specifically mitochondrial ROS here, using mitosox, which lights up red. As you can see, the normal tumor cell, not much red. When you silence it, a lot more red. Erastin is actually an inducer of ferroptosis that leads to mitochondrial ROS. There's even less there than there is in the SI-ABCB7. And then ferrostatin is actually a rescue agent of ferroptosis. So it actually detoxifies those lipid peroxides. And the point of doing this is a rescue experiment is to show that ferroptosis is actually taking place. So we got step 2 as well. And then step 3 is lipid peroxidation. And this red versus green really looks at the reduced species of fat within the cell versus the oxidized fat. What did I say? That the ROS components within the cell are actually targeting the lipid bilayer. So you should see a lot more green, right? So this is a cancer cell, not much green, but when you silence ABCB7, a lot more green. And we did the same rescue experiments and showed that we can reverse it by using a ferroptosis inhibitor. Okay, so at this point, inhibiting ABCB7 elicits ferroptosis. And what did I say? That should actually reduce tumor proliferation. So if that's the case, we should be able to show that tumor burden goes down in a mouse model of medulloblastoma generated from cells that don't have ABCB7. So that was the next experiment. So what we did was we generated 2 sets of mice. Okay, one set of mice had wild-type tumors where we injected these group 3 tumor cells, normal, into their cerebellum, and we saw robust tumors form. And the other group actually had cells wherein ABCB7 was knocked out, and those had expectedly smaller tumors. And so we actually quantified that all the way out to a month, and you can see at day 28 and day 30, those mice that had tumors generated from cancer cells that didn't have ABCB7 had much smaller tumors. And we report here that that's because ABCB7 is lower. So here we're staining the tumors for ABCB7 with a nice brown stain showing high ABCB7 in normal wild-type mice, but our knockout mice, much less signal. So that's how it's happening. And to prove that it's from ferroptosis, this is actually a marker of ferroptosis, 4-HNE, which is one of those lipid molecules that we can assay, and a lot more brown, suggesting that ferroptosis is in fact active in these tumors and reducing the tumor burden. So that's all well and good, but I can't actually go to the clinic or go to Don and say, hey, I'm going to knock out ABCB7 in my patient, because that is a highly toxic thing to do. We have ABCB7 in every single cell in our body, and that iron transport is critical to every cell in our body. So how do we leverage this? Well, could we potentiate classical chemotherapy by somehow knocking down ABCB7? That's the first question, and we'll get to how to knock it down in the next slide. So we asked the question of whether repressing ABCB7 can potentiate cisplatin, the thought being we could probably give lower doses of cisplatin, get the same amount of kill. And so here what we discovered excitingly was that cisplatin actually inhibits ABCB7, and this is not known. What we think about cisplatin is this platinum-containing agent that intercalates within DNA and breaks down the cell because it can't actually replicate. But in fact, it's also reducing ABCB7. This is a novel finding. So then what we did was we had cells wherein we knocked out the ABCB7, and we looked at the dose of cisplatin that would be needed to kill those cells. And we found that when we knock down ABCB7, we need half the dose of cisplatin to kill those cells, suggesting that, yeah, they're working together. This is a colony formation assay. It's a very common assay that's done to look at how well a single colony of cancer cells can grow and establish itself. And in a wild-type situation, they grow beautifully. When you knock it down, you get about a 50% reduction with just knocking down ABCB7. Cisplatin seems to have the same effect, but when you combine them, there's not a single colony in that plate, suggesting that, yes, together they're working to kill these cancers very efficiently. But really the money experiment is going into the mice. So back into the mice we went. We had, again, wild-type tumors generated with normal group 3 medulloblastoma cells. We had another group where we treated them with a dose of cisplatin that's effective in killing those cancer cells. Then we generated mice wherein the cancer cells had ABCB7 knocked out, and then added cisplatin to that group. So 4 different groups. And here's the data on the bottom. What you can see, this is the Kaplan-Meier curve looking at survival, is that the wild-type mice, they die as expected about day 18. If you treat them with cisplatin, they should get better, right? They should die later because that's what we use in group 3 tumors, right, to treat them. And that is recapitulated here. But what's really exciting is if you knocked ABCB7 on its own without cisplatin, those mice are living longer. And when you combine them with cisplatin, I think about 30% of those mice live to about day 44. And so here we were proving at the bench that this combination therapy is going to work. But I just mentioned that you can't practically do that. We need a practical way of targeting ABCB7. So here comes artesunate. So artesunate is an anti-malarial drug that actually is used to treat severe and cerebral malaria. It does so by inducing iron overload. So now we're like, okay, we may have an agent that's FDA approved that we can give to children that may potentiate the effect of cisplatin. We have to test that theory. So first we showed that if you just treat mice that have regular group 3 tumors with artesunate alone, without cisplatin, that you can actually see a reduction in the tumor volume at day 27, just with artesunate alone. I didn't show the data here, but those mice did live longer, but it was a modest increase in their survival. And I'm not suggesting that we start using anti-malarial drugs to replace chemotherapy. I'm suggesting that this might be a way to potentiate the chemotherapy. And so that was the next experiment we did. This is a synergy finder experiment where we treat medulloblastoma cells with different doses of artesunate and different doses of cisplatin in combination, and we see whether they potentiate each other. So red suggests that they are potentiating each other. And so, in fact, what artesunate is doing is it's synergizing with cisplatin to kill these cancer cells. And that's depicted right here in the cell death analysis. So here in green, you see control cells that have a certain amount of cell death. They survive quite well. When you add cisplatin or you add artesunate, they have about the same amount of cell death. When you add them together, you can see that there's a potentiation in the cell death when you add artesunate and cisplatin together. And again, back to that colony formation assay that I showed you a slide ago, the same thing is recapitulated when you add cisplatin and artesunate to these cells. They are unable to make any colonies. And excitingly, we discovered that artesunate, which induces iron overload, can also inhibit ABCB7 similar to cisplatin, which would make sense for a mechanism of how this drug is potentiating cisplatin. And so the next step right now is to really take this model into the mice like we had done in the past. And if that holds out, then we could potentially conceive a phase 1 clinical trial. So let me bring us back to the beginning, to the objectives, to revisit them. First, we've discussed the fact that pediatric brain tumors are the leading cause of cancer-related mortality in the U.S., and that Nebraska ranks 7th in the incidence of pediatric brain tumors. That the incidence of pediatric tumors within Nebraska follows a geographical distribution coinciding with our water source. That the Pediatric Brain Tumor Collaborative at UNMC is trying to bridge the existing gaps in pediatric brain tumor research, and that potentially targeting iron homeostasis using artesunate may provide a novel strategy to potentiate cisplatin action in treating aggressive brain tumors. I want to end by acknowledgments. So very important to say thank you to the people that have been behind this work. And as you can see, it's not, again, within my own lab, there's team science, right? So multiple members within my lab. I want to thank my postdocs and instructors. Of course, my mentors, Dr. Batra, who's the chair of the Biochemistry department, Dr. Mahapatra out in Colorado, and Dr. McComb-Smith at Toledo, who were all early mentors for me when I started this work. Can't do this work without my sponsors, Dr. Kari Simonson from the Department of Pediatrics, as well as Dr. Tucker from the division, who have helped me procure some protected time to do the work that we've done, Dr. Anderson Berry's support in the CHR, and of course my good friend Don, who started me off by giving me my first grant through the PCRG back in 2016. Members of the PBTC that have been essential to the team sciences approach that we have showcased here, and the different grant funding mechanisms, especially the Team Jack Foundation. And before I end, I wanted to give a quick plug to a conference that we're having. So every year, the Pediatric Cancer Research Group puts together a symposium. We are very honored this year for this year's symposium to showcase pediatric brain tumors, the team science that we're doing, and how we can, again, move the needle to help these patients that we're taking care of. Very excitingly, we have 3 excellent speakers that are coming to our Brain Tumor Consortium. So that's Dr. Wetchler-Rea from Columbia, Dr. Rajiv Vibhakar from Colorado, and Dr. Mateos from CHOP. Dr. Rea has made the first group 3 medulloblastoma mouse model, and he's graciously shared it with my lab and the PBTC. As I mentioned, Dr. Vibhakar, who's the chair of the Neuro-Oncology group at Colorado, is my ex-mentor, now collaborator and friend. And Mateos is important and so gracious because at CHOP, they actually have a tumor core facility that's been there for about 10 years, and we hope to replicate their model. They're sharing very nice reagents with us, such as organoids and other such things that we can make available to our investigators. And really the point of this consortium, not just to talk about how we're working on brain tumors, but really to establish connections with these leaders in the field that can help our very dedicated clinicians and researchers move their work forward. With that, I'd like to thank you all for your attention. I've left quite a bit of time for questions, and I'd like to invite Don up here to field those questions with me. Thank you very much for your time.
Any questions from the room? Nothing from the chat yet.
So I'll fill the silence by kind of discussing a little bit about the work that we do with the Team Jack Foundation. So Team Jack has been with us really from the start. They attend all of our symposia. They've been really gracious as far as funding for multiple members of the PBTC as well as beyond. If I'm not mistaken, it used to be called the Team Jack Brain Tumor Foundation. It's now just called the Team Jack Foundation. And they don't just fund work within Nebraska, but really within the United States, the greater United States. And they've got some amazing work that's going on in the coasts at MD Anderson, and they're really gracious partners. They have an annual symposium that takes place, the Team Jack Gala that happens in Lincoln. And when we go there, it's really heartwarming to see the families there. The patients really brings it home to why we're doing what we're doing, and they showcase a patient story every year, and that always moves me because it shows the grit and the incredible tenacity of these children that have these devastating tumors and go through rounds and rounds of chemo radiation therapy and take so much out of them, and yet they're up there kind of still celebrating their life and teaching us so much in the bargain. It's been just heartwarming for me to be a part of that as an intensivist. And really the way I got into this work is because we were taking care of, more brain tumors here in my first 2 years than I'd ever done in fellowship back in the West Coast. And it didn't make sense until Don showed us the work that he did. And it finally all came home, and it decided that that's kind of where I'd like to put my life's work. So it's been a real privilege. Yes, question in the back.
Yeah, so you're talking about ABCB7 and suppression of ABCC7 and getting the kind of synergy between the medications. What sorts of side effects would you conceive of in a clinical trial that you would see from knocking that out?
Yeah, very insightful question. And just to make the clarification here, what we're trying to do is not to knock it out whole body, but really to give it in a targeted fashion, so that things like artesunate that cross the blood-brain barrier will really target those cells that are high producers of ABCB7. But you bring a good question up. Well, what about the liver? What about red blood cells, right? So it's going to be the same side effect profile as we would look at for the anti-malarial drug in a malarial setting. So looking at potential for hemolysis, anemia, potential for LFTs that may change. And those would be the major toxicities that I could think of. But in general, in any clinical trial, what you're going to be doing is looking at a slew of different labs to make sure that we're not missing anything. I should mention that as we are at the precipice of jumping into animal studies, we're also doing pharmacokinetic, pharmacodynamic, and maximum tolerated dose studies in these mice through a collaborator, Dr. DJ Murray. And so what he does is he ramps up the dose of artesunate in these mice until they can't tolerate it anymore, whether that's through weight loss or they look really sick. Once we have the maximum tolerated dose, what we do is we go in and we section different organs at the time of sacrifice. And we look at those different organs in the mice to see what's going on in the liver, in the kidneys, in the lungs, in the brain, tissues like that, to see if there's any evidence of cell death or toxicity there. On the side, then, we run things like CBCs, chemistries on these mice and look at the different panels to see if the drug that we're giving at the doses that we're giving are inducing any of those side effects. And that's how we inform the phase 1 clinical trial that can come after. Now, mice and children are obviously very, very different, but it can give us some insight into which pathways and which organs might be affected.
The question is so insightful that it requires two different people to answer it. The other side of the coin that I think is really, really critical is understanding what the side effects of cisplatin are right now and what the dose limiting factors are, because there are a number of these children that will survive their brain tumors. Afterwards, if they are deaf, which is an incredibly common side effect of the doses of cisplatin that we use currently, that's a lifelong issue that they have to deal with. Or if they have renal toxicity, where they have renal insufficiency or renal failure, which can also be an issue with cisplatin. That can be something that can limit the type of medicines they can receive in the future. So the wonderful thing about using something that acts synergistically is that if we deal with reactions to artemisinin that are blood problems, we can give them a blood transfusion. If there are LFTs problems, we can sort of adjust the dose. But if it allows us to use a dose of cisplatin that is less and allows us not to have as many people have hearing difficulties or renal toxicities, that's a huge win for this patient population.
From the chat, what do you do to protect your own children with the concerns of environmental factors influencing pediatric brain tumors in Nebraska?
Sid, I'm going to let you answer. No. So it's a wonderful, wonderful question. And I want to make sure that we understand a couple of things here first. We are not saying that there's causality. We are saying that there is more research that needs to be done to understand the interplay of the environment with our patients and the development of pediatric tumors. The second thing that we should say is that nobody really knows what causes pediatric cancer. We know of two causes of pediatric cancer that have been confirmed. One is ionizing radiation, and that's been confirmed by when we drop bombs on Hiroshima and Nagasaki. And the second is pediatric oncologists. We actually cause pediatric cancer with the therapies that we give that can cause secondary malignancies. Outside of that, nobody knows. But we do believe there is an interplay between a genetic predisposition and some type of environmental factor. And that is based on data that shows that as countries industrialize, incidences of pediatric cancer increase. It's also sort of something that we hypothesize based on the aggressiveness of tumors here in Nebraska. So there has to be a genetic predisposition, and people have to be exposed to those types of environmental factors for a long period of time. So that's a long way of saying, do I myself limit the amount of water that I drink from Nebraska? No, because I don't know. I don't know. And I live in a urban place where our water doesn't come from a well per se. Our water is filtered and controlled by our municipality. The people that I get really, really concerned about are actually the farmers and ranchers that are so critical to the economic engine that is Nebraska. But on the flip side of the coin, those are the people that are exposed to these compounds. And if they were to develop a pediatric cancer, those families are impacted more by the financial toxicities, the distance from centers where they're treated, and those types of things. So for me personally, I don't do anything different except have a reverse osmosis system in my home. Reverse osmosis removes approximately 95% of the heavy metals and other chemicals that are in water sources. And some of the legislation that we're using in the state of Nebraska currently, while we're trying to understand what is going on with nitrate and other compounds, is allowing for tax breaks for people to be able to use reverse osmosis systems in their own homes. So I want to be very clear. I'm not raising alarms. I'm just raising an idea that science needs to be done in a very, very specific way to help us understand these things. And if I could, I just want to point out the difference between an MD and somebody who has a Ph.D. And you can understand that by watching Sid go through his slides and the scientific method that he employs of having positive controls and negative controls in his experiments and taking those very, very crucial steps before he says, this is something that we should pursue, that's the wonderful thing that happens to somebody when they go through the absolute beating that it takes to get a Ph.D. And that's the type of science that is so critical that we can link together with team science, of taking somebody who is a clinician and working with people, whether they're Ph.D.s in the College of Public Health, to look at the possible environmental factors, or Ph.D.s who are looking at these incredibly intricate systems within cells, so that we can validate data that we're sharing and not just raise alarm bells or raise hopes about something that might conceivably be something we can use without understanding how we could use it.
That was very gracious of you, Don. I really appreciate that. And on the other side, I'm going to say that I can have all the ideas in the world, but if there isn't any funding for those ideas or support from the incredible sponsors and mentors, those ideas are not going to go anywhere. So the team science really requires, I think, not just the scientists and the clinicians, but also a nice network of mentors and sponsors to help them. And really, you know, the presentation that Don showed in the beginning, those are slides that I saw earlier, and I've been seeing those slides change, you know, year after year, and I haven't, you know, closed shop and bounced. So, again, he's right. It isn't causality. It is an important question to explore. And I don't even have a reverse osmosis system. We drink tap water. But again, we test it. If that's one thing to figure out what's in the water. We're cognizant about these things. But I don't think we have something here that we can point to yet that may be linked to these tumors, but we are definitely studying it. We're being encouraged to study it, and it's incredibly important work that I think we need to do to answer the very question that was brought up a minute ago.
Two more questions from the chat. Fenton reactions are significant contributors to iron loss in children by causing GI bleeding. Did you note any new bleeding sites during the mice studies of artesunate?
Excellent question. At this point, no. However, I will say the dosing that we give mice and the dosing we give children are going to be very different. So we have to do that dose escalation experiment to answer that very question. Is that one of the toxicities that we are going to see? So at this stage, with the doses that we're using, we haven't seen it. And you may actually ask the question, how do we come up with those doses? So again, artesunate is FDA approved. There is good PK/PD data in other mouse models, and that's how we start. That's a place to start. But we wanted to do it in our own hands, in our own mice, in our own models, to come up with a much more exact dosing and hopefully be able to answer that question that was brought up.
How can pediatric physicians get more involved in advocacy for families in higher risk areas? Are there programs for water filtration, education in rural communities for families?
Thank you so much for asking the question. I think sometimes people think that there's something special about advocates. There's something different about someone who gets involved in the community and in the legislature. And I can tell you there's absolutely nothing special about me. I am different, but it ain't because of that. It's really, really important that we start with what we see and with the people that are around us. That passion for the patients that you see in your clinic is what's going to allow you to be able to advocate. Then the next step that I think is critical is that our lawmakers, either on a county level or on a state level, are inherently interested in what is going on in our children. We are honored to live in a state that puts our children first for a number of the things that they go through in childhood. And there are a number of philanthropic groups that can help you with any of the things that you're advocating for. But it's important to look at what you see. If you remember, my project started just because of an idea that, my goodness, it seems like I'm seeing a lot of patients from this specific area and going down that way. I think all too often we limit ourselves. We are the first person to tell us, oh, you can't do that. Oh, you're not enough, or you don't know enough. I think the first thing that you should do is silence that voice. And if you have an interest and a passion in something that your patients are going through, advocate for it. This pathway that we went through to develop the Pediatric Cancer Research Group, this can be replicated for a number of the issues that our children face. And I promise you, senators, county leaders, other governmental leaders, they're going to want to hear from you because you are the experts. And you're the ones that are going to show them the needs that we have in our community. Everybody could be an advocate, and it is a key factor of your career in pediatrics.
Anything else from the room? We're good on the chat.
Okay, very good. Well, thank you all very much for your time on this Friday morning. Hope you guys have a fantastic weekend. Don, thank you so much for this opportunity as well.